So today we have an opportunity for the ALS community to learn more about the dexpramipexole trial. The ALS community recently learned that the much-awaited for trial EMPOWER showed negative results. EMPOWER was a phase 3 trial for dexpramipexole, which enrolled 943 people with ALS globally. There was, of course, much anticipation and hope for success because of encouraging results seen in the phase 2 trial. We are very pleased, however, to have with us today Dr. Doug Kerr, the Director of Neurodegeneration Clinical Research at Biogen IDEC, who is going to discuss with us the outcomes of the trial and really give us the opportunity to understand more about what we will learn from this trial and the follow-up efforts of Biogen. So it's my real pleasure, Dr. Kerr, to have you here today, and we will ask a series of questions to get a better understanding of the EMPOWER outcomes.
Thanks for having me, Lucy.
Our pleasure. So our first question really is that in the release that came out, it was stated that the primary and secondary endpoints were not met. Can you describe what went into that process and who was involved? And can you tell us what measures were taken to ensure that this is indeed the case?
Yeah, so first let me just say that we are tremendously disappointed for the ALS community. Everybody in the community had high hopes for this. And we as a company, put a lot of effort into testing whether dexpramipexole works in ALS. And, we did a very good study, and the ALS community should really be commended for being able to do such a good study. But in the end, it's very clear this drug doesn't work. So we looked at multiple measures of efficacy, and those include functional decline, they include survival, they include respiratory status, as well as a series of other measures. And on none of them was there even a hint of efficacy of the drug. And in other words, the placebo group on all of those measures looked no different from the dexpramipexole group. So it's tremendously disappointing. It was an unequivocal finding, however. I mean, it was very clear, and to some degree that's an advantage to the community that we tested in a very sophisticated and very rapid way whether this drug works in ALS, and we just definitively showed that it did not. I've been seeing and taking care of ALS patients for the last 15 years, and I know we want something, but I think that there was a lot of good that came from this study, and we'll talk about that, in a few minutes. But I think it's really important for us to just note that although this was negative and this was a huge disappointment, it really has set the stage for what I think will be brighter days ahead in ALS.
So just following up then directly with that, I think that's a surprise to many because, of course, the anticipation was that the phase 2 results had been so exciting. And in November 2011, there was a release of that phase 2 trial data, which was very promising. So can you talk a bit about how the phase 3 trial may have differed, if it did, from phase 2, and why you would explain these different results? And then also following from that, is it uncommon for phase 2 and phase 3 results to be so different?
Yeah, so, I think the bottom line answer to that is that the phase 2 study of DEX in ALS was intriguing, but it really was a very small study, and it was really not definitive in any way. And we knew that given those results, we had to do the definitive study to assess whether the drug works in ALS. And the phase 3 study was just that. It showed that the phase 2 results were an anomaly. I think that's very important. That's why we do the phase 3 studies. That's why, although the phase 2 results were intriguing, we all knew that they could be wrong. And indeed, they were wrong. The populations were the same. So the drug was the same. The way people took the drug was the same. As we went back at the completion of the phase 3 and said, well, were they different in any way from the phase 2? in terms of, say, the time that they've had ALS, or the rapidity of their ALS progression, or the percent of men or women or bulbar, those types of things, there really was no difference between the phase 2 and the phase 3. They were identical populations. It just turns out that the phase 2 study was not the right conclusion, and we've gotten the right conclusion from the phase 3 study.
And certainly in other clinical areas, I know that they have often said phase 2 are not predictive. What are your thoughts, Dr. Kerr, in terms of is this often the case that phase 2 might well be different from phase 3?
Yes, in drug development in general, that is a huge problem, is that there are these kind of small phase 2 studies, and it turns out that the phase 3 study doesn't confirm those findings. And that happens across all disease areas. But it's an issue that we in the ALS community have to wrestle with now, right? So we need to come together as a community to figure out how can we make phase 2 studies more predictive. And I think over the next couple months, that's what we need to do, is to figure out, maybe the studies need to be a little bit bigger, maybe they need to be a little bit longer, they need to build in some biomarker readouts and things like that will give us greater confidence as to whether or not a drug is working and therefore should go into phase 3.
Absolutely, and as you indicate, that's clearly the positive thing that comes out from these things, because there's clearly already a lot of talk in the community of how to improve this, and we'll highlight that again a little bit later. Will participants in the study receive their individual results?
They will. We feel a deep obligation to the patients and their families who have committed an awful lot to this study, and they need to know what group they were on and whether they were on placebo or dex, and they will be notified very quickly. I should just take that opportunity to really talk about the commitment and sacrifice of patients in the ALS community and how dedicated they are to advancing clinical research, not necessarily for themselves, but for those who will follow. And it really is incredible to see that. The Empower study was a challenging study. It required an awful lot of patients and their caregivers. And yet the patients in the study were amazing, and their compliance with the drug, their compliance with study visits was really something. And so we feel a deep obligation to this community. We're going to continue to develop drugs with them, and we feel that we have to let them know what their allocation was, and that will happen over the next couple months.
Excellent. And I echo your sentiment. I really do encourage, although this was a negative trial, that individuals always, if they can, get involved in clinical trials, because it really will help move the field forward. You have stated that the Envision extension trial will phase out based on these trial results. Can you provide more information for what that timeline will be? Will those taking dexpramipexole need to taper off the drug over a period of time?
Yeah, so that study, the open label extension study, is being terminated now. And the way we have done that is we have contacted the investigating physicians at each of the sites. We've gone over in detail the data with them, and we have asked them to call their patients and bring them in for a follow-up visit to terminate the drug, and then they ultimately will need a safety follow-up visit as well. So that is being done as we speak, and I think at this point, most of the patients are actually off of the drug at this point. We looked very carefully as to whether there could be small subgroups within the ALS population who were treated with DEX and had an apparent benefit, and we could not find that. So, as part of our attempt just to find any hint of efficacy with the drug, we looked at, these various subgroups, subtypes of ALS, and older folks versus younger folks, and rapid progressors versus low progressors. And we just didn't see any hint, and that was why we decided that the open label extension had to stop and patients had to terminate the study.
So in the phase 2 trial, the suggested safety profile was excellent for the treatment of ALS, and it was low risk. Is this something that persisted? It was safe?
Yeah, every drug has some risks, and Dex is no different. But those risks were identified even going into the phase 3 study, and we haven't found any new risk by completing the phase 3 study. So it's just the known risks that we had identified for patients coming into that study.
Excellent. Do you intend to publish the trial results and or make them available to both the research community and the general public?
Absolutely. So the results will be presented at the American Academy of Neurology in March. We are currently writing up a publication so that the study results can be made public to the research community. And we are also very much aware that the clinical database is really a treasure trove for ALS researchers to understand the natural history of this disease better. And we really want to work very carefully with the ALS community to make that available so that we can get the most benefit out of this study that we can.
So the follow-up question here, I think you have addressed, but I'm going to restate it because I think it really does emphasize this interesting question. The comment is, some people say that ALS may be a group of smaller, slightly different diseases, that one reason trials fail may be because the trial is looking at a too large a group rather than smaller subsets. So did Biogen consider this when analyzing the results of the phase 3 trial? I believe you answered that. Was there any evidence of efficacy in a particular subset or differences in the results among subsets of people? And perhaps you could just re-emphasize what you had said earlier.
Yeah, so we looked at various subsets of patients for whether there would be any effectiveness of the drug in those subsets. And really, that would have been a small victory, right? I mean, the ALS community needs a drug, and even if that drug happened to work for only a small percentage of ALS patients, that would have been a small victory, and we could then work to build upon that. And it turned out not to be the case. So that was, again, extremely disappointing in how convincingly negative this study was and how the drug didn't seem to work for any subgroups. But, that's, I think, just the beginning of understanding the heterogeneity of ALS. You and I have talked about this many times in the past, that different ALS patients probably have different mechanisms by which the ALS is progressing. And they may not all be the same. And I think we're just at the very beginning in understanding that. But one could imagine that you could almost use a molecular biomarker to understand why certain ALS patients are different from other ALS patients and develop specific therapies for those subsets of patients. And I think that's probably where the ALS community will need to go in the future.
So I concur with that exactly, and many have asked me as well in the last few weeks, so why don't we just divide up the groups? But I think something that we do have to be realistic about is that it's not as simple as saying, well, let's just do a bulbar versus limb onset or slow progressors versus fast progressors, because I think within each of the subsets there's, again, quite a mixture. Some of them might be bulbar with fast progressing and bulbar with slow progressing. So I think this is going to be the interesting thing and leads on to the next question as to how we're actually going to design these trials, and what do you feel we have learned from this particular trial which will benefit us further, and in particular, again, trial design, perhaps issues on managing a large global trial. What are your thoughts about the things that you really feel we can learn from this trial?
Yeah, I think that's a very important point. I mean, one of the things that I think comes from this experience is that the ALS community can very rapidly do an extremely high-quality study. I mean, so as you said, there were 943 patients enrolled into this study. They all enrolled over a period of 5 months. And that really is extraordinary. And the quality of the data that comes out of this study is extremely high. So we should all feel very confident that we as a community can do really good ALS studies. Now that will be very encouraging to companies who have an interest in ALS and otherwise might not have pursued this drug in ALS, but now will, because we could really rapidly test whether a drug works. There are other things that I think are really very promising. So, for example, the pivotal endpoint in this study is a combined analysis of function and survival, and it was used for the first time in this study, has never before been used in ALS studies, and really combines function and survival in a statistically appropriate but also a very meaningful way. So it's a powerful endpoint that I think is going to be quite important in interrogating whether a drug works in ALS in the future. So I think those are two significant advances for the field. You know, the community through this study, but in parallel with the study, has also developed a staging system for ALS where it really understands how patients progress through their disease in terms of stages, 1, 2, 3, 4, and 5. And I think that is a very important concept in ALS in defining how the disease progresses. I think it's helpful as a physician who would talk to a patient and could communicate to them where in their disease they are. But I also think it gives us a tool to investigate where a drug works. Does it work in the early stages of the disease when patients are independent and employed, or does the drug potentially work in the later stages of the disease? And I think that's meaningful and really needs to be applied in future studies. I think one of the other aspects that was positive from this study is something that we've all been frustrated by for a long period of time, and that is that the time to diagnose somebody with ALS is far too long. And in some cases, it is 12 months. In some cases, it is 18 months from symptom onset until the diagnosis of ALS is given. And the problem is that may, in some cases, be half of the patient's disease course. So it may be, that the disease has progressed too far, that it may be more difficult to treat by the time they actually get the diagnosis. And so we as a community have spent the last couple of years trying to develop a strategy to shorten that time to diagnosis. And it is really now being unveiled, and it turns out that there are a series of kind of red flags or warning signs that non-ALS neurologists should now appreciate as being worrisome for ALS. And what it may do is get patients referred to an ALS neurologist more quickly, rather than going through a somewhat circuitous route that often doesn't get you further toward the correct diagnosis. So I think all of those initiatives tell us that we're at the beginning of what I think will be a bright future for ALS, and that we could really do very good studies. We are increasingly understanding the heterogeneity of ALS. We have very good endpoints. We can get to patients earlier. And so, I see this as the beginning.
Excellent. And maybe you could lead on, if you can say just a few words about the main goals of your new ALS research consortium.
That's right. So we have several initiatives that are underway. One of them is an ALS consortium with many academic centers. And what we've done is partnered with really outstanding basic scientists and have gotten them interested in and focusing on ALS. And specifically, through their academic laboratories and in a collaborative spirit, they will identify new targets for ALS. So we have some good targets in ALS, but there are others that we have yet to identify. And bringing these large academic groups to really put all of their brain power toward ALS, I think is a powerful effort, and I think it will lead to new targets. And then what we at Biogen can do is to go from that target, meaning that specific entity which is causing the ALS, and we at Biogen can say, now how can we modify that? How can we develop a therapy that modifies that target and may slow or alter the disease course? So that's one of the things that we're doing, and that's an ALS consortium that is underway. There's another effort that is to sequence the DNA, the entire genome of 1,000 ALS patients. And that, too, is underway. And what we think that will do is to help us understand the heterogeneity of ALS. We want to know why each ALS patient is distinct from another patient. And they may share some common gene abnormalities. There may be some new causative genes for ALS that we identify from this. But what we ultimately think is it may be able to give us some sense for relevant subgroups of ALS that can then be specifically enrolled into future studies that are targeting a specific pathway.
That's tremendous. It's really exciting to see those initiatives going forward. And I strongly feel if we really are able to better validate the targets before they go into the phase of doing larger clinical trials, I think we also may strengthen our potential. So I guess a question that everyone will ask and really has a complicated answer perhaps, but why has ALS proven to be such a difficult disease to treat and cure?
Yeah, I would love to hear your answer to that question too, Lucy, because you've been such a leader in this field for a long time, so you have to answer that one as well. You know, we knew it was going to be challenging. I mean, Biogen moved into the ALS space 3 years ago. We knew that there had been a string of failures in late-stage clinical trials, and we knew that we were going to experience some failures as well. And it's, you know, for a lot of reasons, right? We don't really understand whether a target is validated. And so we're kind of forced into late-stage studies without really knowing if that's a target that we should be going after. And, you know, I think you said it, that if we can build up some biomarkers to assess whether a target is relevant in ALS disease progression earlier in clinical development, you know, after 50 or 100 patients rather than after 1,000 patients, it helps us to make better decisions earlier in the course. And if it's not working, then you call that off and you move on to the next candidate. And I think that's part of it. I also think, as we talked earlier, that it's the disease may be treatable or more treatable earlier in the course. But after 15 or 18 months of this process, it may be that there are too many pathways that are altered or damaged or causing this disease progression that you can't really stop it by that point. But maybe at an earlier point, it was treatable. And so you can kind of imagine a little spark and a little forest fire that then becomes too big and out of control, and it then becomes very difficult to treat. And then I think the last thing is, you know, it may be ultimately that no single therapy can markedly slow disease progression, but that by the time we actually see the clinical presentation of ALS, that there are so many pathways that are activated that you have to go at it with multiple different therapies in combination at once. And, you know, there are examples for this throughout medicine, right? So if you look at oncology, there's a series of drugs that individually had very little effect, but when placed in combination with one another, now can have some very dramatic effects. The same thing with HIV and the single therapy approaches to halt HIV were largely ineffective. And yet when used in combination, you can now really fully expect complete elimination of HIV replication. And so I think that may be a model that we need to move to in ALS. What's your answer to that question?
I think you've said it beautifully, and I'm not going to try and add too much to that, only to say, of course, if you also think of the system, and I always like to think of things visually, and we show these images of how these complex connections are working, it might be considered focal in that it starts in one area and spreads, but the systems involved in the disease are certainly very complex, with the wiring of the neurons, the motor neurons affected, their connection with the muscle, the surrounding cells. So I think you're already working with a very complex integrated system that, as you say, if it's already gone down a certain path, so many different things are involved that trying to target just one area is going to be a challenge. So I think you really stated beautifully that it is very likely that ALS will be treated by multiple approaches and that each might have an impact and the combination a more significant one on survival. So I'm so encouraged, I think, by the dedication of teams like yourself. Your work in the field has also been a lengthy one in understanding the biology, and I think as we get closer to better understanding the targets and identifying better targets, I think we will be more successful. So I really want to thank you, Dr. Kerr, for a very thoughtful discussion, for some very clear answers that I know will help the ALS community. And most importantly, I want to thank Biogen and the teams there for their continued commitment and for having invested so prudently and so well into teams who are going to help you move this forward and find ultimately better treatments for ALS. So I'm very grateful to you and your team. Thank you so much, Dr. Kerr.
Well, thank you, Lucy, but I also need to thank you and ALSA because for many years you at ALSA have had the vision that we really need to develop the clinical infrastructure, and you have really been a leader in establishing that infrastructure that has made these trials possible. You very much have led the field in developing validated targets and biomarkers so that we can make better decisions in ALS. And so really the fruits of those labors are just beginning to be realized here. And I think it's all because of what you've put in place over the last 10 years that make this so.
Well, thank you. And I think as a team we're committed to this and we're very excited to see the things that are moving forward and, again, encourage participation in clinical trials and being sure that we do further this and we make a difference for those living with the disease. So thank you so much for your time, and we look forward to seeing the progress in the upcoming months and years for ALS.
Thanks so much.